Welcome everyone to today's OTS webinar with Dr. Annabel Biskans, who is working at AstraZeneca at the moment, has recently moved from the core overlap, and she'll be telling us about designing siRNAs, and it should be very interesting because I've seen her talk before. Over to you.
Hopefully. Thank you very much, Petra, for your kind introduction. So I'm really happy to be here online with you to discuss how we can design siRNA to improve siRNA drugs in clinics. So over the past few years, the design of the siRNA have moved from minimally modified and formulated siRNA to formulated base approaches, and now to conjugated base approach, which are actually the best approach to deliver siRNA to a specific tissue. So why the first siRNA drugs get approved in 2018? This drug consists in a minimally modified siRNA encapsulated in LNP, nanoparticles. But as I mentioned, about today, great interest have been to conjugate siRNA to really deliver the siRNA to a specific tissue. And over the last 2 years, already 3 drugs have been approved to treat liver diseases. So I will talk about more how we can design this conjugated base siRNA and how we can achieve robust silencing to develop siRNA in clinic. So if we take, for example, givosiran, so the second approved siRNA drug to treat acute hepatic porphyria, this drug show incredible silencing in vivo, and you can see here that after a single injection, the silencing can be maintained up to 1 month, and even multiple injection can be maintained even after 6 months. So this demonstrates that givosirans, this kind of drug, so this conjugated siRNA drugs, can really achieve an important duration of effect of the compound. So the question here is how we can design the siRNA to achieve this robust duration of effect. And if you look at the design of this givosiran, we can see is mainly composed in different parts. So the first part consists of a fully chemically modified siRNA conjugated to a specific conjugate, here a multivalent GalNAc attached to a pyrrolidine base linker. So I will try to demonstrate how we can design the different part of the siRNA to really improve the siRNA efficacy and to develop robust siRNA in clinic. So first of all, when we look at the siRNA chemical configuration, so as I mentioned, you have really an evolution of the chemical scaffold evolution here. So at the beginning, we were working with unmodified siRNA, and after we moved to partially modified siRNA, and now we are using fully chemical siRNA, stabilized siRNA. So why that? Why chemical modification matters? So to answer to that question, as an example here, different siRNA having different chemical modification content have been evaluated. So on the left, you can see a partially modified siRNA having 60% chemical modification, and on the right side, a fully chemical stabilized siRNA, meaning that this siRNA is totally chemically modified. Both siRNA were conjugated to a cholesterol ligand. So after injection of this siRNA and evaluation of this siRNA, why the chemical modification does not prevent loading or risk activity? It has been observed that having a fully chemically stabilized siRNA is really important to enhance tissue accumulation compared to a partially modified siRNA. You can see here in red, when you use a fully stabilized siRNA, in tissue after a systemic injection, so liver, kidney, and spleen, we can really see that a large amount of compound accumulate on those tissues compared to the partially modified siRNA. So in order to deliver siRNA to tissues, a fully chemically stabilized siRNA is really essential for systemic delivery. So this is one of the reasons why we need a fully stabilized siRNA, and so why we need to modify siRNA. Another point that I want to make here is that this enhancement of tissue accumulation of the siRNA is not dependent on the conjugate attached to the siRNA, either of the route of administration. For example, in the context of cholesterol conjugates, DHA conjugate of GalNAc conjugates, either in IV or after IV or sub-Q injection, this enhancement of accumulation was observed in all of the cases using the fully stabilized siRNA. Compared to when you use a partially modified siRNA, you can see almost no tissue distribution, no tissue exposure at all. So this is to make the point that we need to stabilize the siRNA to use these conjugated base approaches to deliver efficiently siRNA to tissue. So to stabilize this siRNA, to do that, we can use chemical modification. And so a lot of chemical modification have been explored over the past decades, especially explored in the context of antisense oligonucleotide. But so here's a few examples. So you can see you have modification. You can modify the backbone of the siRNA by having a phosphorothioate, for example. You can also modify the 5' phosphate of your oligonucleotide, or your ribose. You can, for example, substitute your 2' hydroxide by chemical modification. So in theory, all this modification can be introduced in siRNA. However, you have to keep in mind that these chemical modifications have been developed and explored in the context of antisense oligonucleotide that does not require risk loading. So some of this modification may interfere with the risk loading of your siRNA. So some of this modification cannot be used in the context of siRNA. So at the moment in the clinic, 4 modifications have been commonly used. So the phosphorothioate modification for the backbone chemistry, the vinyl phosphonate to stabilize the 5' phosphate, and the 2' methyl and 2' furrow modification to modify the ribose. So these modifications have been introduced in siRNA in clinic and show that they cannot interfere with the risk loading of the siRNA. However, the point I want to make, take me slow, further is like even if this modification can be used to stabilize siRNA, you have really to be careful when you design your siRNA where you place this modification within the duplex and how much of this modification you introduce within the duplex, because it can really impact your activity of your siRNA. As an example here, it's a really nice paper from Einaylam, which has been published in 2018, and they demonstrate that when you change only one chemical modification in one specific position, you can inhibit your siRNA activity. So here they compare different chemical patterns where they replace a 2' furrow modification by a 2' O-methyl modification in position 2, 6, or 14 of the antisense or 11 of the sense strand. And by doing that, they observe that the siRNA is not efficacious anymore. So this demonstrates that having a more sterically demanding modification of specific position is really detrimental for efficacy. So you can use chemical modification, but you are really careful when you design your siRNA to place this modification in a right position. So they also demonstrate in the same paper that having more 2' methyl modification when you carefully position this modification within the siRNA, you can increase the potency of the siRNA. And here, for example, they evaluate the potency of two different siRNA, one siRNA having 50% 2' furrow and one siRNA having only 20% of 2' furrow. So they increase the O-methyl content of the siRNA. And what they can see by doing that, by increasing this 2' O-methyl content, they were able to induce a better silencing and a more prolonged effect of the siRNA. So what is important here when you design? So you have to stabilize the siRNA by using chemical modification, but you have to be really careful about the position and the content of the chemical modification. And it seems like having a rich siRNA having 2' O-methyl modification is more beneficial to develop robust siRNA in clinic. So in addition to the chemical modifications contents, also, a stabilization of the siRNA is really required to achieve this robust silencing in tissue. And to demonstrate that, here is two examples of two different compounds developed by Anailam, so revusiran and givosiran. So revusiran have been developed a few years ago, but have been discontinued because this compound show an imbalance in mortality between the drug-treated patient and the placebo group. At contrary, givosiran is an approved drug, like I mentioned previously, to treat acute hepatic porphyria. So both of these two drugs have 2' O-methyl 2' furrow modifications. So why one was discontinued, one was really efficacious and is now used, is now approved. So if we look more closely, if you look of this pattern, we can see like in the revusiran, they don't have, this compound doesn't have 5' stabilization of the strand. In contrary, in givosiran, you can see here in red, they have this phosphorothioate modification. So meaning you stabilize more your duplex. So in this context of no 5' stabilization and 50% 2' O-methyl modification, revusiran was injected at a dose of 25 grams per year per patient, which is reduced. It's a really large dose. So this can explain the toxicity observed with this compound. So, at contrary, in givosiran, so you have this 5' stabilization of the strand, as well as more 2' O-methyl content, so 55% of 2' O-methyl. So as I mentioned previously, having a more O-methyl content of in your siRNA allows a better silencing, better durational effect. So this is an improvement of the design. And you can see as well that at the 5' end of the antisense strand, you have a vinyl phosphonate to stabilize this 5' phosphate. So why we need a 5' phosphate in the first place? So this 5' phosphate is really necessary on the 5' and the antisense strand to be able to load the siRNA into the RISC. So normally when you use and modify siRNA, the siRNA go inside the cells, and the 5' position, so of the antisense strand, get phosphorylated by a kinase inside the cells, and so the siRNA get loading into RISC. However, when you totally modify your siRNA, your strands become a poor substrate to this kinase, and so the phosphorylation of this 5' end is limited. So you limit your RISC loading. You limit the siRNA enter to the RISC. So to avoid that, you can introduce chemically a 5' phosphate, but however, when you do that, this 5' phosphate is not stable enough in tissue to be maintained, and so you will lose this 5' phosphate, and so you will decrease your RISC, your siRNA loading into the RISC. So a lot of groups, labs and company have been working to stabilize this 5' phosphate using analogs. So these analogs is just a few examples here. So you can use, for example, an analog that instead of having bridging oxygen here, you can replace by a carbon. You can substitute as well in the 5' position with a methyl. You can use a phosphorothioate, or you can use this evinyl phosphonate here. And what is really interesting with this kind of modification is that they stabilize this 5' end of the antisense thread, but they also enhance the interaction with the agoprotein into the risk, so they enhance the capability of the siRNA to be loading into risk. And what has been demonstrated using this 5' stabilization is like, for example, here, if you use a vinyl phosphonate instead of 5' phosphates, or instead of 5' OH, no phosphate at all, you can increase your activity of your siRNA in vivo. So this show that having this kind of stabilization is really important to maximize siRNA performance. So having all these kind of stabilization, so the 5' stabilization in 0 end of the strand, 55% of 2' of methyl, and having this vinyl phosphonate allows to decrease the dose less than 1 gram per year per patient. So only by designing very well the siRNA, so by stabilizing the siRNA, we went from 20 gram per year per patient to 1 gram per year per patient. So the difference is huge. So this show how much is important to really well design and modify the right to put the right modification into your siRNA. So the next question here is, okay, so it's really important to stabilize the siRNA. But if you stabilize even further the siRNA, can we improve even further the activity of the siRNA? So what have been done here is like the content of phosphorothioate chemical modification have been increased. So moving from 8 phosphorothioate modification in total to 13 phosphorothioate modification. And so the goal is to understand the impact of the content of phosphorothioate modification on siRNA distribution and efficacy. And the goal is to see if we can increase the stability, we can increase more the activity a bit further. So after injection into mice of these two compounds systematically, what has been observed is that the phosphorothioate content does not impact much siRNA accumulation in tissue, but it does impact silencing. And it has been demonstrated that having a high phosphorothioate content is detrimental for efficacy. So meaning that it's really nice, really good to have chemical modification to stabilize your siRNA to be able to achieve robust silencing in tissue. But if you induce too much of one kind of modification, for example, phosphorothioate, you really impact siRNA activity and you will lose some activity. So these results can be due to the fact that phosphorothioate content, we know that it can interact with different protein, and so having a lot of phosphorothioate in your design can change your interaction with protein inside the cells and can just change your activity of your siRNA. So this is another example that's why you have to really be careful when you design your siRNA. When you add your chemical modification, you need to choose which modification, which position, and how much you want to include in your siRNA. Another point I would like also to mention is that, okay, chemical modification are crucial to be designed in a careful way, but also the structure of the siRNA is really important. So here you have an example of different structures that have been explored with different labs and company, and you can see that, for example, Alnylam use this structure where they have the 2 nucleotide overhang at the 3' end of the antisense strand. But some companies use also overhangs of both ends of the siRNA. Some use blunt compounds, however, some use dicer siRNA having this tetra loop into their structure. So I will not go into detail of all the structure, but I will just give one example to show you how this structure is really important when you design your siRNA. So you have to choose the right structure according your conjugate you have and your tissue you target. So here, as an example, 3 different siRNA having 3 different structures have been evaluated. So 3 of them are a fully stabilized siRNA having lipids as a conjugate. And the difference between these two structures is like having either an overhang, so 5 nucleotide overhang or 2 nucleotide overhang, or not overhang at all. And so the goal of this study was to see if the siRNA structure impact siRNA tissue distribution and efficacy in tissue after systemic administration. So when we look at the data here, we saw that having an overhang or not an overhang does not impact siRNA accumulation in extrahepatic tissue in the context of lipid conjugated siRNA. However, it does impact activity, and we have observed that siRNA having an overhang induce better silencing than blunt siRNA, again in this context. So this shows that other than the chemical pattern of the siRNA, the structure of the siRNA is really important when you design siRNA. So upstanding chemical scaffolds show robust, efficient, and prolonged efficacy due to the right optimization of the chemical composition, the structure of the siRNA, and the content of the chemical composition of the siRNA. However, we can wonder if this chemical stabilized siRNA will be toxic because they're not very heavy chemically modified. So we can ask how maybe this modification can impact the toxicity, and we can increase toxicity in tissue. So I will talk about 3 points here. So we can worry like the 2' furrow modification induce toxicity. Indeed, like a few years ago, Ionis published some papers where they demonstrate that 2' furrow modification can be toxic in the context of the antisense oligonucleotide. However, this kind of toxicity induced due to the 2' furrow has never been demonstrated in the context of double-stranding siRNA. So the reason that is that because having a chemically stabilized siRNA, using this kind of compound, the dose you use to treat patient is really low. So the risk to see toxicity due to chemical modification is really limited. But with that said, we have to be careful to see if this kind of modification impacts, induce some toxicity at long term. So we don't know that yet. So it's maybe something to look to watch for in the future. Another effect that can induce toxicity is the off-target effect. So when you use a siRNA compound, you can target your gene of interest, but also it's possible that you target another gene, so you reduce the expression of the protein, so you induce toxicity. So to avoid this off-target effect, we have tools. We have bioinformatic tools. So when you design your siRNA, you sequence, you make sure that your sequence that you selected it will not interact with another's genome in your cell of interest. We have also we can also use chemical modification to reduce this off-target effect. And Anailam published a beautiful paper demonstrating that if you introduce a specific chemical modification as of position 7 of your antisense strand, such as GNA modification, you can destabilize the pairing in the seed region, and so you decrease your off-target effect. So this is really something, of course, always when you design a siRNA, you have to be careful of this off-target effect because it's the most common sign of toxicity. So you have to watch for that, but we have tools to decrease this. Another point I want to make here is we have seen that using this chemically stabilized siRNA, we can induce a duration of FFA up to 6 months, even 1 year after injection. So this durability of the compound have been investigated by Anilam, and they published a beautiful paper in 2020 that showed that in the context of GalNAc siRNA conjugate, this siRNA can accumulate into an acidic intracellular compartments and act like a depot to really be able to load the siRNA to risk and to help and to have this long duration of effect. So we can have a concern here, maybe because if the risk is if you continuously load your risk, you can interfere with mRNA homeostasis. However, it's never been shown in the context of siRNA. And the risk of that is really low because when you load your risk using this GalNAc siRNA, you load less than 5% of your risk complex available in the cells. So it's unlikely that the toxicity due to this depot in the cells will be observed. So, so for now, I would say the chemical stabilized siRNA developed in clinic looks pretty safe. Okay, so we have seen that fully chemically modified siRNA is really important to design to increase efficacy of your siRNA in tissue. But what about also the conjugates? It's another part of the siRNA and is as important of the siRNA chemical configuration. So can we design this conjugate to increase the siRNA activity? So we have seen like in Giroziran and in the other drugs I've proved so far, the GalNAc, the multivalent GalNAc conjugate have been used. And this GalNAc chemistry has really revolutionized the field for liver disease, to treat liver diseases. It's incredible how it's really efficient to deliver siRNA to the liver. However, it's not one way to conjugate to put the GalNAc attached to the siRNA. It's a multiple way to attach GalNAc, and you can have a multivalent GalNAc, a cluster GalNAc. You can have also monomer GalNAc in series, or you can have different number of GalNAc at different position, 5' end of the sense strand, 3' end of the sense strand, etc. So a lot of labs and companies are exploring the design of the GalNAc to see if we can really improve even better the siRNA activity in liver. So here is really some few examples. I will not go through all of them because it's a lot in the literature, but again, I will show an example. So this data is from Silence Therapeutics. I published a nice paper last year in 2020. And what they do in the papers, they screen different GalNAc conjugated siRNA, where they change the position of the GalNAc and the number of the GalNAc moiety attached to the siRNA. And they demonstrate that it does matter. Like you can change the position, you can affect the efficacy as well as the numbers. So this is an example to show that even if GalNAc is really nice to deliver siRNA to liver, we can still optimize this to even achieve a better silencing in liver. So now what about the other ligands? Because GalNAc can be used to deliver siRNA to liver only. So there have been a lot of efforts from academics lab company to try to discover different other ligands than GalNAc that target different tissue. So here's some example. You can use peptide conjugate siRNA. So AstraZeneca, in collaboration with Ionis, show beautiful data when they use the GLP-1 peptide to deliver specifically oligonucleotide into pancreatic cells. Santirine conjugate have been also developed by Arrow Biotherapeutics, and they show that the santirine conjugate can really bind tightly with a specific protein in different cell type. Antibody conjugated also have been developed to deliver siRNA to muscle. Avidity Bioscience and Dyne Therapeutics show really nice data about that. So I really recommend to watch their presentation on that. And a lot of papers also have been published using Atamer conjugate, where they show really promising data to use this strategy to deliver oligonucleotide to a specific cell type and tissue. So other than this targeting moiety, you have also a lipophilic conjugate to be able to deliver functional siRNA into extrahepatic tissue. So I will talk a little bit more in detail of this lipophilic conjugate because it was my main postdoctoral project in Anastasia Vorova's lab. So what we have done to understand if we can design in a particular way the lipids to really achieve functional delivery in extrahepatic tissue here. So we have designed different lipid conjugates with distinct physicochemical properties. So conjugation of different classes of conjugate, fatty acid, sterol, vitamins, incorporation of polar groups such as phosphocholine group, or modulation of the valency of the conjugates. So I've been explored to understand the relationship between the structure of the conjugate and the PK/PD property of the siRNA. So the different lipid conjugates have been attached of the fully stabilized siRNA, and they have been injected into mice via a single subQ injection. And after 2 days post-injection, so spatial distribution and antisense strength quantification in 15 tissue have been performed, and later on, some silencing efficacy have been done in order to see if we can deliver efficiently this conjugate in several tissues. So first of all, when we look at the distribution of the different lipid conjugated siRNA in liver and kidney, what we observe is that by only changing the chemical structure of the conjugate, we can alter the liver-kidney distribution. And we observe a correlation between the hydrophobicity of the conjugate and the liver-kidney distribution. Less hydrophobic conjugate, as hydrophobic compound, accumulates more in kidney than liver, and high hydrophobic compound accumulates more in liver than kidney. So this difference in liver-kidney distribution can be explained in part by the capability of the siRNA to bind spontaneously with different lipoprotein in serum. In general, high hydrophobic compound bind with LDL and less hydrophobic compound bind with HDL. So explaining why tissue, for example, rich in LDL receptors such as liver tend to accumulate more high hydrophobic siRNA, such as cholesterol siRNA, for example. So this was really interesting to see that by only changing the chemical structure of the conjugate can be designed and really engineered to achieve predictable partitioning into lipid transport pathway, and liver-kidney distribution and clearance kinetic can be predicted. So while the nature of the conjugate, the hydrophobicity of the conjugate drive the overall distribution in kidney and liver, we observe a really interesting thing. It's like the structure of the conjugate impacts the cell distribution within the tissue. Here, for example, in kidney, if we look at EPA conjugated siRNA and PC-EPA conjugated siRNA, we can see that these two conjugated siRNA accumulate to the stem extend to the kidney. But in using EPA siRNA, we can deliver siRNA to glomerulus cells, where when we use PC-EPA, we don't. So by only having this phosphocholine polar group in the conjugate, we inhibit this distribution in this specific cell type. So this is really to show you that you can really design your conjugate to really tune your distribution within the tissue. The same was observed in the liver. So cholesterol, PCTS, and DC conjugated siRNA as an overall tissue distribution in liver, but they have different cell distribution profile. Cholesterol accumulates everywhere and mostly in hepatocyte cells, but DC siRNA accumulate only in Kupffer cells and endothelial cells. So we look also at other tissues than liver and kidney. We look at extrahepatic, extrarenal tissue, and we have demonstrated that also we can deliver efficiently conjugated siRNA in different tissue, heart, muscle, lung, and fat, for example, or we can reach significant silencing on those tissues. So again, it's just to make the point that by engineering and designing your conjugates, so having a saturated or unsaturated fatty acids, having a phosphocholine group, so a polar group, or changing the balancing of the conjugate, you can modulate your distribution of the siRNA tissue to be able to achieve functional silencing in those tissues. So so we have seen that both the siRNA can be engineered, can be designed to maximize siRNA efficacy, as well as the conjugates. But what about the linker between the conjugate and the siRNA? Can be also designed in a certain way to improve siRNA activity. So in the context of GalNAc, in the Givoserant here, a specific linker have been used, the pyrrolidine-base linker, and so we can ask if it's the best linker to improve siRNA, to achieve potent silencing. So what have been published 3 years ago now, is like they evaluated different linker chemistry using this GalNAc conjugate, and they used two different approaches. The cluster-based approach of the GalNAc, that means the GalNAc, it just have this multivalency, so these three GalNAc attached to some linker, and this have attached to the siRNA. Or they also use a monomer approach where the GalNAc have been attached in series along the siRNA. And they study different linker using these two different approaches. So the aniline linker, the pyrrolidine-based linker, and a linker having TEG spicer. And for the monomer approach, they use either a phosphodiester linker or a TEG spicer as well. When these different compounds have been injected into mice via systemic administration, what they observed first is that by changing the chemical structure of the linker using this cluster GalNAc, it doesn't impact much the activity of the siRNA. At high dose, you can see between the red and the light blue a slightly improvement of your efficacy using the aniline linker, let's call it like that. But because the GalNAc is so potent, you don't see a huge gap of improvement of efficacy when you change your linker. They also observe, like having this monomer approach, you can decrease the silencing, the activity of the siRNA compared to the cluster approach, especially at high dose, if you compare the red and the dark blue here. So this show that in this example, of course, you can change your linker chemistry and you will impact slightly your siRNA activity, but because the GalNAc is so potent, you will not see maybe too much change when you change the linker, the chemistry of your linker in the GalNAc context. But what about in lipid conjugated siRNA? Because the silencing is not as good at the GalNAc, so maybe here we will see a more like impact of the chemistry of the linker of siRNA activity. And yes, it is a case. So when two different siRNA, lipid siRNA, have been evaluated into mice via sub-Q injection, they compare here a stable linker and a cleavable linker between the conjugates, the lipids, and the siRNA, and they have been demonstrated that having a cleavable linker is really beneficial for siRNA activity. So this means that the linker can really be optimized, designed in a way to improve siRNA activity, and it seems like in this context of lipid conjugated siRNA, a cleavable linker is preferred. So this can be mainly due to the fact if you cleave your ligand to your siRNA inside the cells, you can maybe improve your endosomal escape, and so you can improve your siRNA activity. Okay, so to conclude, so you have in the past decades, a lot of effort have been done to really design and optimize each part of the siRNA. So the chemical composition, the chemical content, the linker chemistry, and the conjugate to really achieve robust silencing in tissue. There is a lot of room of improvement here, of course. It's not perfect. We can still optimize even more the siRNA. And the first thing I think we need to understand is how these siRNA interact intracellularly with different proteins to really understand which part of the assay we have to really optimize further. So I will stop there. So thank you very much, and I will be happy to take any questions. Okay.
Is this going to be available online later? Yes, it will be. Yes, Annabelle has agreed to make the PDF available. How do you deal with the stereoisomers, RPS or SPS? Do you select purify the stereoisomers with high activity?
So in the context, it's true in the context of antisense oligonucleotide to have this stereoisomer RPS or SPS dramatically change your activity and the stability of your antisense. So in the context of siRNA, it has been tested where the S isomer has been proved to have better stability. But the advantage in the context of siRNA to have this stereoisomer compound compared to the isomer, the PS, normal PS, does not improve much activity. So in the context of siRNA, I don't think it's necessary to use this stereoisomer. So do you select to increase? I mean, you have to be more worked on that, of course, because it's always a balance between stability and activity, right? So if you use one stereoisomer that stabilizes your siRNA but is detrimental for activity, and you use the other isomer, it's more active, but it changes stability. It's a little bit tricky. But in the context of siRNA, it's not that important. With SO oligonucleotide, I would say yes. I don't know if I answered.
We have another question from Fabrice Leclerc. How many phosphorothioate modifications can be incorporated without any significant toxicity in terms of numbers or ratio modified and unmodified?
So in terms of toxicity, I don't know. In terms of efficacy, as I mentioned, the optimal scaffold is to have like these two phosphorothioate at each end, as a 5' end of the antisense strand. So to achieve the efficacy, we need to really understand which protein binding interaction, so the PS content in the context of siRNA we have, because we know in the context of antisense oligonucleotide, but not in context of siRNA.
Michaela Denti would like to know the data on toxicity of 2' fluorometabolites. I think you already referred to the Janus paper from Al-Nalam on that, so I think that contains all of their data.
Yeah.
Any known off-target effect associated with the 5' phosphate modifications used for stabilization?
So yes, of course, when you stabilize your 5' phosphates, so the goal is to load into your siRNA, but you want to load your antisense strand. So you have been some work when you stabilize this 5' phosphate. You have to introduce a chemical modification that do not interfere with AGO binding, AGO2 binding. If you do that, for example, instead of E-vinyl phosphonate, you introduce a Z-vinyl phosphonate, so other configuration, you will not load your antisense strand, so you can have off-target effect by loading your sense strands.
Okay, where is GalNAc located? At 3' terminus of sense strand instead of the 5' terminus?
So as I mentioned, it's a different way to conjugate the GalNAc to siRNA, so mostly on the sense strand, so 3', 5', and the 3' end of the antisense strand. So what you want to avoid is the 5' of your antisense strand, because you want this strand phosphorylated to be able to load the strand into the risk. But so depends of the company, depend lab, so you have different position where you can conjugate the GalNAc.
And then Neges Asriz, in most of your works from Professor Anastasia Kroer's lab, the length of siRNA is 2020. Why not 21?
Yes, I mean, Anilam used 23. We use 20. You have to be between, so you don't want something too short, because if you're too short, you will have a lot of unspecific effects. So you want to be between 19 and 25. If it's too long, you will have a lot of off-target effect, and it will recognize as well as your immune response will be stimulated, so you will have toxicity. So you have to be on this 19-25 length. So 20 is on the range. You can use 23 if you want. It's not like set rule.
Yi Yin would like to know that if you have one structure showed give us a run with VP modification, but the other showed no VP, can you confirm if VP modification has been used in the approved siRNA drug givosiran?
Yes, yeah, I confirm that.
How are siRNAs formulated in general?
Formulated, can the person can specify, mean like in nanoparticles, or
I imagine for the injection in mice, perhaps.
So if you just inject without any nanoparticle, you just dilute your siRNA PBS, for example, at certain concentration, and you inject directly the mice with your solution. So you don't need to formulate it, because you have this the siRNA is chemically modified, so it's stable. It will not be degraded by enzyme, and you have the conjugate that help to improve the half time of the distribution of the tissue of the conjugate. So you don't need any formulated agent.
What are these new tools for off targets you mentioned?
So for the off target, I mentioned the bioinformatic tools. So this is more blast tools, or you can have so basically you have your sequence, and you can identify if your sequence target other gene in your genome and your 5' UTR in your cells. So this can be one tools. The other tool I mentioned here is to introduce chemical modification in specific position, and ILAM show that having this destabilization of this of the pairing in the seed region of the 7th position of the antisense strand allow to decrease this the off-target effect. So these are the two tools you can use to decrease that.
Yeah, there's also a very recent paper. I'll post a link that is about one of those tools that is free, made freely available.
Thanks.
Is there any software for asymmetric siRNA design?
Available online, not yet. So when we were using this asymmetric siRNA in the Kforova lab, the lab designed the tools, the software, based on. So basically, we screen a lot of asymmetric siRNA, and we set up some rules, and the algorithm have been built on that. So at the moment, it's not available online, if I'm not mistaken.
Michaela Denti, I assume most of the rules you have talked about apply for siRNAs delivered through intravenous injection. Is there something different in toxicity, efficacy, efficiency, and so on when injecting into the CNS?
Subcutaneous injection versus IV injection is the same rules. So the difference, you will have not the same pharmacokinetics of your compound. I mean, when you do IV injection, your compound will go thoroughly in the blood, so it will distribute more quickly in your tissue. So, this rule can apply for both, I will say. For the CNS, yes, it's mostly you inject directly in the brain. In terms of toxicity, we don't see any difference. I recommend to read the really nice paper in biotech technology that Anastasia Labs publish on this divalent siRNA, or they show like the toxicity using this stabilized, fully stabilized siRNA is not an issue at all, and you can have an efficacy up to 6 months in certain region of the brain. So I don't see the issue in CNS using the stabilized chemical siRNA.
There's a comment, I guess, by Jan Weiler. Inclisiran does not carry a VP and is working great. Yes, it is, but...
Yes, I mean, it's a different way to design siRNA, right? You can put other modification. People are investigated to have different, instead of phosphorothioate, no, it's maybe the PS2, or it can be something else. Of course, you have different way to stabilize siRNA. So I just mentioned the VP because it's something we know and it's something can be important. But I think if you find a way to stabilize, another way to stabilize the siRNA is working as well.
David Slack would like to talk about while GalNAc targets liver and companies like Avidity and Dyne are targeting neurological and muscle tissues. What approaches show promise for delivery to solid tumor cancers?
I think they also show in tumor that antibodies approaches can be used because this company use antibodies for muscle delivery, and they show really nice data. But if you engineer your antibody to target solid tumor cancer, you can target this cancer as well. So I will suggest maybe to look a little bit more on what IVT is doing on that.
Valentina Fankia, how all these modification influence encapsulation efficacy? You don't encapsulate at all.
Yeah, you don't exactly. You don't encapsulate because you don't need to. If you want to, I mean, because the goal of to have all this modification is not to use a nanoparticle, because you want really avoid you want to deliver your siRNA different tissues and liver, for example. So when you encapsule, it's hard to really deliver to siRNA into a pretty specific tissue other than liver and kidney. But I don't see why this chemical modification will impact encapsulation, because they're not something will interact with the membrane. And we will see, like in patisirants, they have some modification. It's not fully chemically modified, but they have some modification. The siRNA have some modification using the LNP. So I don't see any influence on that.
I think the list of questions is just getting longer and longer. We've got a question here about a company that can produce docosahexaenoic acid conjugation and the VP modification. I'm not sure that's an appropriate answer here, because we're not endorsing any of the companies.
Exactly, yeah.
I would just look at some of the top companies that produce oligonucleotides, and if they don't have it on the website, just please contact them and ask. They're usually able to accommodate if you have the money to pay for it.
Yes. Thank you, Petra.
Did you evaluate the administration via inhalation, for example, CNS delivery? Can you tell about the way RNAs are prepared and administered?
Inhalations. Yes, we did some work in inhalation during the COVID time, because we were working on COVID siRNA, like siRNA targeting the mRNA of the virus. So you doesn't need to prepare like another way as you do for systemic administration, because you just so depends what you call by inhalation. If it's just like a intratracheal administration, you don't need anything. If you want to use like some nebulizer, I'm not an expert at all on this field, so I guess you have to prepare in a way, like you have to do some like powder, maybe dry powder of your siRNA, but I'm not sure. I'm not an expert at all in this field, so I don't know that.
How many siRNAs are in the clinic that do not use a delivery system, and what is the toxicity? I think that's quite a question here.
A lot in clinic. It's a lot of clinical try using GalNAc siRNA. I don't know the number of more than 20, 30. I don't know the number. And regarding the toxicity, I mean, for those have been accepted, so have been approved either by the FDA or the European Union. So it's 3 of them using the conjugated base strategy, and they show no toxicity at all. So, but the other that are in clinic, maybe it's too soon to see if there are some tox associated to that. Yeah, I don't know that.
I would like to say that, well, Alnylam, Sikana, there's quite a few companies who have things in clinical trials, so it's worthwhile to look at their websites and check out the listings that they have of their what they have in testing. What is the most effective, efficient approach behind conjugation chemistry? Could you please provide examples?
So you can conjugate in different ways. So you can use solid supports that already have your ligand on it and grow your oligonucleotide on it. So this is one way. You can also use a phosphoramidite strategy, or use a phosphoramidite to put your ligands on your siRNA. But this has to be it's a bit tricky, because the chemistry of your conjugate have to be really adaptable with the condition that you use during the synthesis. And the other way is to do post-synthetic reaction. So you can attach your conjugate after you in solution when you have your siRNA. So you do you can do click or NHS ester strategy to conjugate that. The most efficient approach, I would say, I like to use the ones that use like the conjugate on the CPG, because when you do post-synthetic reaction, it's not total, it's not perfect. So using this CPG with your ligand on it, at the end when you purify, you're sure that you have 100% of your siRNAs that is conjugated. But it's different way. As an example, I mean, all the lipids that I show, like what we did in Anastasia lab, we use conjugate, that we use CPG with the ligand on it. But in the GalNAc, like a lot of companies such as Silence, for example, Therapeutics, they use the monomeric approach, where they use the phosphoramidite. So I think it depends on what you like the most. And yeah, I think it's not like a better or the worst, but you have to make sure. So it depends what you want to conjugate, because you have to be sure that your ligand is compatible with the chemistry you want to do. So this is the only thing you have to be careful of that.
I think we need to maybe speed up a little bit, because the messages, questions keep piling up. Is it possible that siRNA conjugated with cholesterol may interfere with the LDL system when you systemically administer?
Yes. So using cholesterol conjugated siRNA, this has been a paper in 2006 or 7 in Nature Communication. They show that the cholesterol interfere with LDL lipoprotein. Yes.
Do the lipid siRNA form micelle structures?
No. So we check that, and when you have only one lipid, it doesn't. When you change the valency of your lipid, so when you have three fatty acid, for example, you can see some micelle structure. Yes.
Is a cleavable linker benefit observed in siRNA conjugated to anything other than lipids?
In the literature, so no, because for example, for antibodies, AVD show they don't have any cleavable linker. In the context of a GalNAc, so depends if it's siRNA or of antisense, but siRNA, I mean, Analab has this phosphate, so it can be cleavable, right? So maybe on the GalNAc is also beneficial.
Can you comment a little bit on the administration method for the DISRNA, the work from the Coral lab?
Yes, so it's administered locally directly in the CNS. Yes, it's locally injection. It's not systemic administration. It doesn't cross the brain barrier, so it's directly injected.
Alexandre Debacker, thank you, Annabelle. Amazing talk. I fully agree to that. What would you recommend? What would be your recommended path to develop a fully modified siRNA? Would you start by screening unmodified sequences, then screen different fully modified pattern scaffolds on the working sequences, or would you go from the beginning fully modified siRNAs using your preferred fully modified pattern to identify the best?
Yes, so I will start screening modified siRNA, because we have shown, like, when you use algorithm, if you use one that you can use for unmodified sequence and the one we use for modified sequences, they're not correlate at all. So you have to be careful when you design. When you use the first screen, I will definitely go with the modification first, including new design.
By siRNA structure, you mean sequence? Do you mean sequence?
No, I mean if we have, like, for example, here an asymmetric siRNA, or you have like a blunt, so it's more like the length of the strand is more in that way. The sequence is nothing to do with it.
Yeah. I think we've had that question. Yes, the recording will be available as usual. It is available from the OTS website, as usual. Well, in 2-3 days. Okay. What cleavable linker would you recommend for lipid siRNAs?
So what I've been done here is just having, like, DNA, for example. So it's like what I showed, the result with the cleavable versus non-cleavable edited lipid is just a DNA linker. So I will really recommend that. It's cheap, easy to easy to synthesize. So I will use that.
Any recommendation for macrophage delivery?
I
think I darkly remember an Aptamer delivery on that, but I can't remember the paper.
Yes, I think I'm sure I have the paper on that. It's just have been published to 2020. I think they use as well GalNAc. No, I'm not sure about that. I don't want to book. Sorry, I don't know.
How is the future for oligonucleotides, siRNAs? What's your opinion?
It's we have seen over the past 2 years, we have 3 drugs get approved in clinic, and way more are in the path. Like in the pipeline, is like way more get approved. So I think the future is really promising, and it's really great. It will expand, and we will use this technology for multiple to treat multiple diseases in multiple extrahepatic tissue. So I'm really optimistic about this technology.
Yeah, if you want to find more about oligonucleotides and siRNAs, then please check out the OTS website. I'll post the link in a moment. Is position important for phosphonates? For example, the vinyl phosphonate in sense or antisense strand of the siRNA?
Yes, it is important. I think really soon it will be a paper published from Anastasia group, or they place different phosphonate linkage in different position of the siRNA. So I will invite you to read that paper. It will be out soon.
Do you know if those lipid siRNA conjugates have a misylate? No, we've had this question. Why do you modify the sense strand? Which strand loads into RISC complex? That's so basic.
So so it's the antisense strand is on the RISC. We will load it if you RISC. I mean, both can be loaded, right? It's 2 siRNA, 2 strand. But if you want to target your on-target effects, you have to load the antisense strands that you designed for your target. Yes.
Toxicity of the phosphonate modification of the siRNA?
So depends how many phosphonates, where you put the phosphonate on your siRNA. So depends of your structure. It can be toxic at different position. I mean, or if you put too much, yes, depends, I guess.
Have you ever observed upregulation then rather than knockdown when using certain modifications?
No, never. No, I mean, no. I mean, if you design really well, when you do like the bioinformatics, when you design your sequence, yeah, it should knock down and not upregulate it. So I never observed that, but I guess it can be possible.
Yeah, if you look at if you look at small activating RNAs, particular MENA technologies here in London do this. It's slightly different mechanism, but it's sort of very similar structure to start off with of the RNA.
Yeah, definitely.
Are there any examples of LNA modification siRNAs in clinical trials? Not to my knowledge.
No, not to my knowledge as well. Yeah.
Christopher Brown says that VP is not being used in Give Us a Run.
Oh, okay. Sorry about that. Okay, I thought it was. Thanks.
2' modifications are base or position dependent. 2' modification refers to the 2' sugar.
Yes, exactly. So by 2' modification, it's just you replace your 2' hydroxyl with some modification. So yeah, I'm not sure.
Okay.
If I understand that.
Would you have any bioconjugate recommendations for delivery to skeletal muscle?
So yes, I mean, in the lab we published a paper this year that's using fatty acids, DCA, you can deliver functional siRNA to skeletal muscles. So I invite you to read that.
I would. sorry, someone commented GLUT4 ligands if they exist as a recommendation. What's the percentage of lipid conjugates that will go to other tissues, not to liver and kidneys? With respect to GalNAc conjugates, is the dose you need to get an appreciable silencing much higher?
Yes, this is a good point. When you use lipid conjugates, so depends of your lipids, so depends where you distribute your tissue, your siRNA, sorry, you can reach like 5% of your injecting dose in, for example, using this DCA conjugate that I mentioned before for muscle, you can reach 5% of the injecting dose on this tissue, on muscle tissue. So, and to do that, the dose is much more higher. You need like 10 mg per kg, 20 mg per kg. So, of course, GalNAc is way more efficacious to deliver siRNA to liver. I mean, it's really specific because using a lipid is not a targeting moiety you're using.
Okay, I think we've made it to the last question. Between conjugates and nanoparticles for oligonucleotides, is there a competition between conjugates and nanoparticles for oligonucleotides delivery? Probably each technology is most suitable for a specific purpose.
Yes, I mean, depends, I guess, your tissue you want to target. If you want to target, because having your nanoparticle, you can engineering, I guess, to deliver in different tissue, but you increase the size of your drug, so you cannot reach any tissue. So I agree, it can be complementary here, depending on the tissue you want to target.
Yeah, but also need to point out if you use a nanoparticle, then that has to be produced and tested, and the whole formulation has to go through the whole approval process. So it makes it a bit more expensive and more complicated.
It's true. Yes.
Okay, I think we've reached the end of the questions, and there's lots of thank yous from all the participants.
Well, thank you all. Yeah.
I would like to point out that, yes, the recording will be available soon from the website. I've posted the link on the chat. And I would like to point out that we have the next presentation with Professor Stanley Crook. And he'll tell us all about the N-Lorem Foundation, about N=1 ASO trials and how they're helping patients with that. And that will be on the 9th of February. And again, you can find the details on the Otis website. Thank you very much, Annabelle.
Thank you so much. Yeah, it was a great pleasure.
Thank you very much.
Thank you. Bye-bye. Bye-bye.